So this is the second part of our lecture on photosynthesis. Photosynthesis does have two reactions. The step one is actually a light-dependent reaction because it requires light energy. The step two is what we call our Calvin-Benson cycle. This is going to be our light-independent reaction, or it has also been referred to as the dark reaction because no light is needed here. So photosynthesis does begin when light is absorbed by a photosystem in the light harvesting complexes. These are located in the thylakoid membrane itself. So remember, the thylakoid was the actual little stacks of little pancake looking items inside of the chloroplast. So this is the membrane of the thylakoid itself. Right here, this huge purple thing is the light harvesting complex itself. As you can see right here, because the light harvesting complexes are where the pigments absorb the photons. So photons are the form that light energy travels in. So light energy, those photons, are going to sit here and hit these light harvesting complexes. These are going to go ahead and allow the energy to be absorbed, and then it bounces from the highest right here to the lowest all throughout here. And then from there, it's going to be passed on to the reaction center, which is the center portion right here of the whole complex itself. As you can see, the center portion, which is called the reaction center, has a pair of chlorophyll A molecules. And we're still talking about the light-dependent reaction in this case. But the light-dependent reaction does consist of two photosystems. So you do want to know that photosystem 2 actually gets reacted upon first, and then it goes to photosystem 1. The reason it was actually named this way, photosystem 2, photosystem 1, is only because this is the order that they were discovered in. So the light energy, which travels in the form of photons, comes in and it hits on this light harvesting complex right here. The light energy is going to go ahead and be transferred from one to the next all throughout here. And then finally what's going to happen is it's going to come in here to the reaction center. From here, what's happening is we already had electrons waiting to be shot up, but it needed this energy that came in from the light energy to shoot it to a higher energy level. Well, where the heck did these electrons come from? These electrons came from this water molecule. So water actually got broken down into an oxygen and hydrogen molecules. But remember, when you break a bond, you release energy in the form of electrons. So these electrons have been sitting here waiting, and once that light energy hit, it was enough energy to shoot these guys up the reaction center. And then now it's allowed, because it went from a very high energy level, and it's going to go ahead and come across the electron transport chain where it's passed through different proteins here. So that was going through photosystem 1. Photosystem 2 works very similarly, where you still have photons coming in. That light energy is being passed on through here through the light harvesting complex. Once again, that electron that got shot up from here and made its way down the electron transport chain is now waiting right here where it says P700. When the energy hits, it's going to allow it to be shot up through the reaction center and finally make its final destination through this last ubiquitin protein. So one thing you do want to notice here is that the photosystem 2 actually has a P680 right here, whereas photosystem 1 has 700. This is basically telling us at what wavelength this actual pigment is absorbing those photons at. So photosystem 2 absorbs really well at P680. Photosystem 1 absorbs very well at P700. Some things you do want to notate about the light-dependent reaction is you do want to understand what's going in and what's actually coming out of this reaction or this portion of it. So one thing that came into the reaction portion is water, okay? Another thing that had to come in for this to occur is light energy. So we had water and light energy. And then what's coming out is we actually have, when two of these water molecules are broken down, it creates an oxygen. This, remember, is one of the waste products, or one of the products that comes out of the photosynthesis actual chemical equation. So this is oxygen that's coming out. Not only do we have a release of oxygen, we do have some ATP being made, as well as NADPH. That is one thing I didn't mention about what's coming into this. That has to be here. So we did mention water. We also mentioned the light energy. But one other thing is we need NAD+, because that has to basically combine with this hydrogen due to this electron energy coming through here. And it'll actually go ahead and allow NADPH to be a product of here as well. So taking a look at this, remembering the slide that we just looked at right here, remember photosystem 2 occurs first, then it goes through electron transport chain, then finally through photosystem 1. Remember, you do want to notate what's coming in and what's going out of this part of the light-dependent reaction. So continuing on from what we just looked at in the previous slide, we're going to talk about the cyclic pathway and also the non-cyclic pathway in the next slide. The cyclic pathway only uses photosystem 1. So this guy right here, or in this case, here it is right here in this picture. This was actually a cyclic pathway was evolved and came about before the non-cyclic pathway, and we'll see and explain each of these. So what happens with the cyclic pathway is only photosystem 1 is used. That's why you're only seeing this guy. You're not seeing photosystem 2 and 1 here. And the energy that's released from photosystem 1 is released in the form of electrons. Well, that energy is enough energy to allow hydrogen ions to go ahead and shoot across the thylakoid membrane into the actual inside of the thylakoid, which is called the thylakoid compartment. The high concentration of hydrogen ions inside this area right here actually ends up being a huge concentration gradient, enough so that the actual thylakoid wants to shoot these guys back out to try to go ahead and maintain equilibrium once again. When it shoots it back out, what it's doing is actually going through this enzyme called ATP synthase. So it naturally is the enzyme that's actually going to be creating ATP. We can see it right here from ADP and a phosphate group being phosphorylated, or adding a phosphate group to this guy right here and creating our ATP molecules right here. So electrons at the end of this electron transfer chain go back to the start of the photosystem 1, completing the cycle, and it goes on and on and on. So what would make more ATP or be more productive for a plant rather than just using this one photosystem? Well, naturally, using both photosystems that are available. So the non-cyclic pathway uses both photosystem 2 and photosystem 1. So this is going to be a much more advantageous way for the plants to actually go ahead and absorb as much energy as it can from the two different wavelengths, P680 and P700. And it's also going to allow it to go ahead and create a lot more ATP due to the fact of having a whole lot of electrons coming in from the, like we said, the different wavelengths from photosystem 2 and photosystem 1. And not only that, but they're also able to make a whole bunch more ATP because of how much more energy they can actually create. A lot more energy coming through here, a lot more energy being created right here. And last but not least, that means a higher concentration of gradient of the hydrogen inside the space, therefore resulting in our creation of a lot more molecules of ATP right here. So what we just went over was the light-dependent reaction alone. If you can recall, what does the light-dependent reaction require? Well, it does require light energy and also water. The light-dependent reaction is going to be converting our solar energy, remember our light coming in as photons, into chemical energy or ATP. Where does it take place? You are going to want to know where in the cell it takes place. So the light-dependent reaction does take place in the thylakoid membranes of the chloroplast. It does use photosystem 2 and photosystem 1, and it actually releases or creates oxygen, NADPH, and ATP. So what happens when there's an excess amount of light energy that the plant has to deal with? So in this picture, in the top picture, you're actually seeing what the chlorophylls look like when they're actually been blended together in some water. Down here, you'll see what it looks like when we actually have a UV light directly looking at it or directly in contact with this guy right here. And this is what you're seeing right here. You're actually seeing the fluorescent light emitting heat coming through because there's an excess of energy available. So let's see what we're talking about right here. Remember when that light energy was first coming into the actual light harvesting complex, let's say in photosystem 2, for example? Well, what was happening was those electrons, remember, they're going from a low energy state, and due to the new photons coming in, they're actually jumping to a higher and then higher and then higher energy state till eventually it's too much energy, and then those electrons actually drop back down. They're excited electrons, and then they actually have to drop back down to their original energy level. When they do that, what they're actually going to be doing is getting rid of that extra energy that they basically built up. And that extra energy that they built up, and that's what we know as the emission of fluorescent radiation, aka fluorescent lighting, also the heat that's being released. Because as you can see, light entered at 450 nanometers. It got those electrons really excited. They moved up to their highest energy level. Guess what? Then light has to actually leave at 540 nanometers, so a lot more extra energy than the plant needed for photosynthesis to occur. So there it is right here being released once again as fluorescent radiation. So that was all we just talked about the light-dependent reaction. Now moving forward, here is the light-independent reaction and what you're going to need to know about it. You're going to need to know that can also be called the dark reaction. It's really not done in the dark. They just call it that because there's no light required. Or it can also be called the Calvin-Benson cycle. Here is where carbon fixation is going to occur. It does not require light like we just mentioned. And it's going to be requiring or using ATP and NADPH, which we just got as a product from the previous reaction. So right here, let's take a recap really quickly. Light-dependent reaction. Light energy comes in. It actually hits in the thylakoid membrane. And this is all a chloroplast right here. Okay. So remember, light-dependent reaction occurs in the thylakoid membrane. We had photosystem 1, the electron transport chain, and photosystem 2 that had to occur. And then finally we had the huge influx of hydrogen ion concentrations inside the thylakoid lumen area, or that space in the middle. And that pumped hydrogen ions back out into the stroma area, which is all this like liquid-like fluid on the outside of the thylakoids. And when it did that, it pumped it out through ATP synthase, which actually created ATP. So what went into the light-dependent reaction was water. Also, we had ADP and phosphate available, as well as NAD+. And what actually resulted from the and also light energy. And then what was the result of the light-dependent reaction? We actually had oxygen be created here. And we also had ATP and NADPH be created, or as a product of this cycle. So as we just mentioned, on the actual Calvin-Benson cycle, we do need carbon dioxide. We also are going to need ATP and NADPH, which we are both getting these from this light-dependent reaction. So it's actually going to be this reaction's products, which we're using for this reaction's reactants. Okay. So one thing you're going to want to know or have to know is that Calvin-Benson cycle uses the carbon atoms from carbon dioxide to build carbon backbones of the sugar molecules. So here's our carbon dioxide coming in. So this half right here to the right is all the Calvin-Benson cycle. Once again, it's occurring in the stroma of the chloroplast. Okay, so what happens here is our carbon dioxide coming in. It's going to actually be broken down through a series of reactions, as you can see. So right here, ATP is being broken down into ADP. That is a release of energy. We also have NADPH being broken down into NAD plus and an extra hydrogen ion. When we're coming around this cycle, one thing you want to know, and that's very important, is right here, this part right here. So through this cycle that's occurring, G3P is being created. So if you all recall, glucose is actually a 6-carbon molecule, and it is the smallest of all the monosaccharides, or the smallest of all the carbohydrates that there are that exist. So G3P is actually just a 3-carbon molecule. So in order to get our 6-carbon glucose, we have to go around this cycle 2 times to generate just one glucose molecule. So once again, carbon dioxide comes in. We also need ATP and NADPH, which we can also see right here, as reactants as well. And as it's going through the cycle, it's going to actually be breaking these guys down, each of these, and it creates a G3P molecule, which is just a 3-carbon molecule, which is why it has to go around twice to produce one 6-carbon molecule of glucose. Another thing you want to notate is G3P, which is this molecule down here, regenerates the ribisco, which is being created right here. Ribisco is also one of the items that has to come in when CO2 comes in to go ahead and allow this reaction to, or this cycle to go through over and over and over again. So let's go ahead and look at light independent reaction recap of what we just went over. So it's going to be using carbon dioxide, NADPH, and ATP from the light dependent reactions. It actually generates G3P. G3P is a 3-carbon molecule, so that's why you have to go around the cycle twice to make a 6-carbon glucose molecule. And it also is going to be generating NAD+, ADP, and free inorganic phosphate groups right here. It occurs in the stroma, which is all that liquid-like fluid inside of the chloroplast. And once again, I'm pointing this out because it's important and you are going to have to know it for the exam. It does take 2 complete Calvin cycles to create one glucose molecule itself. So this is actually in detail what's happening, and it does describe in this slide what rubisco actually is. So rubisco is just a plant enzyme that catalyzes the fixing of atmospheric carbon dioxide. So that's why we say carbon fixation occurs here. That's when carbon dioxide is fixed from inorganic to an organic molecule. CO2 enters. After that, it's added in with this rubisco molecule coming in here. Then that actually undergoes the entire cycle through here that we just went over. And remember, this is our 3-carbon G3P molecule, 1, 2, 3 carbons, and that's why it has to go around twice. It regenerates rubisco again, and then it goes through the whole thing once again until it finally makes one full molecule of glucose. Okay, you're not going to have to know these structures. You're not going to have to know this is actually rubisco. You're not going to have to know the structures. Just know what's going in and what's coming out of these actual stages itself right here. Also know where it occurs. And so this is giving you a nice little brief summary of each of these items, where it's being reduced, where carbon is being fixed, which is right here at the very beginning, and then where the regeneration of rubisco is occurring, which is right over here at the end of the cycle. You will want to know the actual definition of photorespiration. It's a wasteful pathway, which you can see on this side right here, that occurs when the Calvin cycle enzyme rubisco that we just talked about acts on oxygen rather than carbon dioxide. Okay, why does this occur or what's actually happening here? So in all reality, rubisco is actually a lot more attracted to oxygen than it is carbon dioxide. Well, we don't want the plant to use up the oxygen that's actually creating, because that would be very wasteful. Not only that, because we won't get the oxygen we want that the plants release as a waste product, because then that's what we use to respirate ourselves. And it is very energy consuming as far as the amount of energy that is actually broken down going through this photorespiration cycle over here. 1 PGA formed and 2 carbon molecule that breaks down to release carbon dioxide. So instead of going through the cycle and fixing carbon like this side did over here, what it's actually going to do is be releasing carbon dioxide as well. So this is a very wasteful pathway for plants to undergo. So why would plants actually have this as an option, or even be able to do a pathway that's so wasteful? Well, the reason being, during times of drought, or depending on where the plant is located, they might have to undergo photorespiration to basically, when they have to close their stomata or their guard cells, because they do not want to allow gas exchange to occur. So you are going to want to know the differences between these kind of plants. Okay, so majority of plants are considered what we call the C3 plants. So they're going to go through exactly the cycle we just went over together, where CO2 comes in, it gets acted upon by the rubisco, and then it's going to come around and around. It breaks down ATP, breaks down NADPH, and then right here we create 1 G3P molecule, and then it regenerates rubisco and then goes around the cycle again. Okay, they have no special features to combat against that wasteful pathway known as photorespiration. C4 plants, however, have C4 plants and CAM plants have a really cool way of avoiding photorespiration if they can. So C4 plants, what they can do is they can avoid photorespiration by separating the initial carbon dioxide fixation of the cycle and the Calvin cycle in spaces, performing the steps in different cell types. So the mesophyll cell can actually do the carbon uptake for us, and then the actual Calvin cycle, where everything gets broken down and we basically need to regenerate the rubisco and all that stuff, that can actually be done in a separate cell known as a bundle sheath cell. That way, the actual plant does not have all that free carbon dioxide just hanging out here for photorespiration to our rubisco to act upon it. So it's a really smart way for plants to avoid photorespiration. CAM plants are also able to do something that basically affects how much photorespiration is occurring. The way that they're able to do it, and remember, photorespiration is very wasteful. They waste water, they waste gas, all that kind of stuff. So the way these guys are able to do it is they actually are going to be separating these steps, the carbon uptake and the full Calvin-Benson cycle, between night and day. That way they only take carbon up during, let's say, the nighttime, but it cannot be acted upon because the Calvin-Benson cycle will only cycle during the daytime. So it's a really good way for these two plants to be able to avoid photorespiration from occurring. This is a really nice labeled slide that you might see in lab, so I wanted to give you guys a visual to look at. So this is a nice little table summarizing what we just went over. Like I said, you're going to want to know what C3 plants do and how do they combat against photorespiration. As we just said, they actually do not have a way to combat against that. Whereas C4 and the CAM plants do have a way to do it. You are going to want to know how they do it. Okay, and you're also going to want to know what kind of plants are best adapted to that kind of plant type. So for example, CAM plants are usually going to be best adapted in very hot and dry environments because they're able to do the actual carbon dioxide uptake and Calvin-Benson cycle at separate times of the day. So a really nice brief summary from beginning to end of some photosynthesis. Remember, we have light energy coming in. That light is going ahead and hitting our chloroplast, inside our chloroplast, we have stacks of thylakoids. So the light energy hits our actual thylakoid membrane, where photosystem 2, the electron transport chain, and photosystem 1 are acting upon. You're going to want to know, remember, what's going in and what's coming out of this cycle. You do need to know that it's occurring in the actual thylakoid membrane. Then you want to know the products that actually go over here into the Calvin-Benson cycle, or the dark light independent reaction over here. Also, what goes in and what are products of this cycle. Where does it occur? It occurs in the stroma of the chloroplast itself. And remember, it does take 2 cycles to actually create just one glucose molecule itself. You all have probably already heard of the greenhouse effect. So what exactly is happening with this greenhouse effect, and how is it affecting us today? So what's happening is, in a normal, and how it normally works is, the sun is what's heating up the earth, which is really, really good for us, because obviously we do need the sun's energy and the sun's heat to go ahead and warm the earth, because without the sun, we could not survive. The thing is, some of the energy is actually reflected back out into space because of the ozone layer that kind of protects us. It gives us a little protection barrier. The other solar energy itself that, travels as photons passes through our atmosphere onto the earth itself. So what happens if all of those bad chemicals or gases that we use here on earth that causes the pollution actually basically builds up our ozone layer thicker and thicker and thicker and thicker? Okay, so why is that an issue, or why is that affecting us? Because as the sun's rays comes in to the earth and passes through our atmosphere, due to our pollution and this huge layer that we're creating, what happens is that sun, remember, it's supposed to be able to emit back out. It's supposed to be able to emit back out and go back out into the space area over here. Well, because of this kind of like little tent that we're creating, the heat is actually stuck in our atmosphere and comes back down. So it's almost the total amount of heat that originally came in, that part of the heat that used to leave actually gets stuck in our atmosphere. That is what's leading to the actual higher temperatures that you're seeing nowadays. We're having record highs in multiple locations. Well, why is that a problem? Because the higher temperatures on Earth means, like for example, those in those what used to be very, very ice cold places, the ice is actually melting. Well, when the ice is melting, number one, the ocean levels rise. As the ocean levels rise, that means that all those beautiful corals that used to use the sun's rays to grow, well, the water is now too deep to reach them. That's why you're having coral bleaching occur, where the entire coral will be wiped out clean and pretty much die off because they can no longer get the heat energy that they need from the sun. Also, I'm sure you've seen a couple pictures online or videos of the polar bears actually, going starving and pretty much dying out, because now they used to live in these ice cold environments where there was actual icebergs for them to live on. Now, with ice melting, they're having less and less area that they can call home as well. So you do want to be able to describe the greenhouse effect in your own words. And here are some questions to think about while you're studying. A plant in extreme water stress from lack of water will not only wilt, but also do what? So you are going to have to think of the photosynthesis reaction and think about which step that actually required water to occur. So if you can recall, water was actually required for the light independent reaction. Remember, water was splitting into the hydrogen ions and oxygen ions. Okay, so with no water, that means we're also going to run out of electrons for the light dependent reactions to occur. Another question you want to think about is the light energy captured by chlorophyll in photosystem 2 goes first to blank and then later to blank. Light energy captured by the chlorophyll in photosystem 2 goes to ATP first, and then later to G3P, because this is a 3-carbon product once again that will be later turned into glucose or other kind of molecules for the plant itself. This is the end of our lecture. Have a great day, and just as always, email me if you have any questions and I'll be happy to help you.